πηγαίνει παρά 20. Αφετέρου, ο κύριος Λεωνάρντι έχει και μία άλλη διαδικτυακή ομιλία στο Παγκόσμιο Συνέδριο των Φλεγμονών. Πρέπει να τον αποδεσμεύσουμε. Λοιπόν, το θέμα του στρογγυλού τραπεζιού αφορά τη φλεγμονή της οφθαλμικής επιφάνειας και εδώ ακριβώς θα μιλήσουμε για διάφορα θέματα που απασχολούν την οφθαλμική επιφάνεια, άλλα περισσότερο κλινικά, άλλα στο χώρο της έρευνας, αλλά είναι όλα ενδιαφέροντα. Αυτό που θα αλλάξει, αλλά δεν προλάβαμε να το κάνουμε, ήταν, και ευτυχώς είχα backup, είναι η ομιλία του κυρίου Νικολάου, η οποία θα αφορούσε σε αποστήματα το cross-linking, τη χρήση του cross-linking και την θερμοκερατοπλαστική. Το πρόβλημα ήταν ότι ο κύριος Νικολάου αντιμετώπισε ένα πολύ σοβαρό προσωπικό θέμα και δυστυχώς και δεν μπόρεσε να έρθει, αλλά και αποδιοργανώθηκε στις τελευταίες μέρες αυτές. Ελπίζω να πάνε όλα καλά, γιατί ήταν ιδιαίτερο σοβαρό. Οπότε, για να καλύψουμε το κενό και για να δέσει κάπως στο στρογγυλό τραπέζι, θα μιλήσω πάνω σε κάτι που αφορά αυτοάνοσα νοσήματα και κάνει περιφερική ελκωτική κερατίτιδα και θα δέσει και με την εισήγηση της κυρίας Τερζίδου, που θα μιλήσει για τον ξηρό οφθαλμό, οπότε ουδέν κακόν να μηγές καλού. Να καλωσορίσω πρώτα απ' όλα τον καθηγητή από το Πανεπιστήμιο της Πάδοβα στο τμήμα των Ευρωεπιστημών. Good afternoon, Professor Leonardi. I enjoy your presence with us, and since you have other obligations in the IOS Congress as well, I will give you as first speaker the opportunity to tell us for a very specific issue and very interesting, what do we learn from gene expression in vernalkeratoconjunctivitis? Please.
Good afternoon. I would like to thank the Atlantic Society for the study of ocular inflammations and infection for this invitation to talk about the gene expression in VKC and what we learn about this study. Ocular allergy is one of the most common inflammatory diseases of the ocular surface that might be associated with rhinitis, but also be a unique presentation in our patient. Particularly in severe cases, we have patients that suffer for visual disturbance, many problems for their activities, tissue remodeling are shown yesterday in my previous presentation. Ocular allergy is a Th2-mediated disease with specific IgE produced, inducing particularly seasonal perennial allergic conjunctivitis, but in part also VKC and AKC, but it's also non-IgE mediated, particularly in VKC and AKC and contact blepharal conjunctivitis, where Th2 effector cells play a major role. Once the patient is sensitized, we can have an acute phase and a late phase, which is typical prolonged reaction, which might induce chronic changes on the ocular surface. As shown yesterday, there are many aspects of allergic inflammation that might be taken in consideration as cofactors and different effects we can find on the ocular surface. Today, I would like to talk about the abnormal gene expression and dysbiosis of the ocular surface, particularly in VKC. VKC is it's a particular form of ocular allergy that for us is a prototype of Th2 type inflammation which can be relatively easily studied to understand the pathophysiology of ocular allergy in general. VKC affects mostly young males, but not only. Resolves at puberty, but not all the time. It's characterized by longer, prolonged, or perennial inflammation associated to other allergic manifestation only in half of our patients. The typical Th2 immunoresponse of the ocular surface is characterized by intense eosinophilic inflammation with Th2 cells and Th2 type cytokines overexpressed, but probably not only that. We believe the Th17 immunoresponse is also involved, and as I said, I showed you yesterday, there are many other involvements such as corneal involvement and tissue nodule that play a role in the history of the disease. We had the opportunity to study gene expression in VKC using conjunctival transcriptome analysis, collecting samples with impressive cytology device from 25 VKC patients and processing these samples for the nanostring technology and analyze the gene expression. So what we found in this VKC patient that 55 of the almost 600 genes that were detected after cleaning and doing the lab work, 55 genes were overexpressed independently from the subtype of VKC, and this is the volcanograph that shows the significantly overexpressed genes compared to the control, and looking at different families of genes overexpressed. There were very interesting family that highlighted to be particularly overexpressed. The first was the host pathogen interaction family, followed by chemokine and cytokine signaling family, innate immune system family, Th2 and Th17 differentiation family, but also the NLR signaling and TLR signaling families. Dividing the VKC patient in the three main phenotypes, tarsal, limbal, and mixed. See that in tarsal there were the more numerous genes significantly overexpressed, less in limbal, and something between for the mixed form of VKC. The tarsal and limbal had 12 different overexpressed genes in common that were related to the inflammatory activity, but also to the immune innate family genes. VKC the gene expression was also correlated with the activity of the disease. That means in more severe patient, more severe with disease, more expressed genes were present or overexpressed, and this was shown by dividing the patient by the Bonini scores, the general activity score. Less evident if we look at just one aspect of the disease, which is the corneal staining by both Oxford and VKC-CLEC scores. In another study, we have shown that autophagy was overexpressed in VKC because we found significantly increased overexpression of autophagic markers in tissues, in VKT tissue in particular, but also in vitro we showed that some of the cytokines involved in ocular allergies, such as TNF-alpha, significantly increased the autophagic flux in the conjunctival fibroblasts, showing that not only autophagy is related to inflammation, but probably also to the remodeling process. So we look at the gene expression for autophagy and apoptosis markers in VKC and found that even in the gene expression, there were several autophagy related genes overexpressed in VKC, but not too many related to apoptosis. So apoptosis is not really activated in VKC. Actually, we believe it's down expressed. But also there was an increased expression of the inflammasome marker and RP3 gene, which is a marker for inflammasome process. What was particularly interesting in our study is that of the overexpressed genes, many were related to the pathway recognition receptors families. So not only TLR, toll-like receptors, but also many other genes that recognize microbe particles. And these are some of the genes that were overexpressed in VKC. Dividing the patient by IgE and non-IgE VKC patient with high score or low score. In any case, we found that genes related to innate immunity family or the host pathogen interaction were overexpressed. They were particularly overexpressed in high score patient, but both in IgE and Ig positive and Ig negative subtype or subgroups of patient. And this overexpression of pathway recognition receptors was proved by immunohistochemistry analysis from conjunctival biopsy, where the toll-like receptors, NOD and 1 and 2, and NL, PR3 and CLEC 4A and 7A and MRC1 were overexpressed. The protein was overexpressed either by the epithelium or in the conjunctival stroma. This scheme highlights the fact that toll-like receptors, in particular toll-like receptor 4, stimulate the different types of immunoresponse. But in our case, the two immunoresponses that were particularly overexpressed in terms of genes were related to the Th2, the family of genes that help the differentiation of the subtype of T cells, but also the Th17. In different study, we look at the microbiome, the local microbiome in VKC, and we found higher microbial alpha and beta diversity in VKC compared to control, and increased bacterial dysbiosis in VKC compared to control, with particular Proteobacteria, Firmicutes, Actinobacteria, Bacteroidetes, that were significantly overexpressed in VKC compared to controls. And these families accounted for 90% of all the reads, which represent the core microbiome. Similarly, we found also a dysbiosis regarding the conjunctival fungal microbiome. Interestingly, Malassezia was particularly significantly different in VKC compared to the controls. So we believe that the overexpression and the redundant expression of multiple pathway recognition receptors, toll-like receptors, NLR, CLR, and ALR receptors on the ocular surface, particularly in VKC, might interact with glycan, phospholipid residues from allergen microbes or other environmental factors that can engage these receptors on epithelial cells or dendritic cells in the conjunctiva, priming directly on the ocular surface a type of a subtype of immune response, particularly in VKC, the Th2 and Th17 response, or trigger directly the inflammatory response. Another study on gene expression on the ocular surface, we have been, in the last 2 years, we've been interested to the expression of SARS-CoV-2 receptor and show the AC2 and proteases was expressed on the cornea and the conjunctival cells, not at a high level. But we were particularly interested by some reports in the literature that showed that asthma might be protective against COVID-19. And surprisingly, patients with asthma are not having a severe disease as expected if they had the COVID SARS-CoV-2 infection. So we look at the gene expression related to the viral response. We were interested because only 2 VKC patients were affected by COVID-19 without any ocular symptoms, and evaluated gene expression of SARS-2 receptors or gene related to the receptors on the conjunctiva in VKC subjects using Affymetrix assay. And this is the genes that were related to the SARS-CoV-2 receptors, AC2 and TMPRSS2, that have been shown to be overexpressed in inflammatory diseases, were not upregulated in VKC. Look at the geoterm defense response to virus. We found 38 genes that were overexpressed in VKC compared to control, with several antiviral factors overexpressed in VKC, suggesting that local persistent allergic inflammation might be protective for viral infection. So we have several evidences that gene expression in VKC in particular can help us to understand the pathophysiology of the disease and probably find alternative treatment for our patient. And thank you for your attention.
Thank you, Professor Leonardi. I have a common question. Can you describe the kind and the level of help that give us the detection of gene expression in clinical practice?
Thank you. That's a very complicated question and not easy to answer. I believe that these studies on gene expression are explaining the variety of phenotypes we can have in VKC. So even though phenotypes are relatively similar, the endotypes, so the pathophysiology of each of them might be different. We have patients that are Ig positive, patients that are G negative, and the disease is, at least the phenotype is the same. However, the underlying pathophysiology is different, because in cases we might have a Th17 or Th2 type inflammation, or even just an inflammasome mediated inflammation. That's explained why some of the patients are allergic to environmental allergen, and some are completely negative. And the exacerbation of the disease is relatively unspecific to a specific environmental condition. So we don't have right now the actual translation from these studies to the clinic, but we might have in the future, maybe identifying additional targets for a therapeutic approach.
Okay, thank you. Last question. The antigen-presenting cells of the surface of the cornea, of the surface of the eye, can be modified by the gene expression. I mean, if we have 4, 3, or different gene expressions, these genes can modify the response of the antigen-presenting cells in cases of allergy, especially in VKC.
Well, thank you. Again, very complicated question, not easy to answer, because probably so far
it's complicated in my mind as well.
Yeah, okay. Well, theoretically, yes, but we don't have the approach with specific therapy to target this possible mechanism. So unfortunately, so far we have no data and no really a way to approach the dendritic cells. Maybe with some kind of vaccination, but it's too early to say that.
Okay, thank you very much. So,
Thank you.
Good evening.